It is my honor to introduce our distinguished speaker, Dr. Yogesh Kumar, an assistant professor at the Department of Data Science, Center of Biomedical Research in Lucknow, India. With over a decade of experience as a PDF scientist, Dr. Yogesh specializes in biological data analysis and drug discovery. Associate editor and reviewer for prominent journals like Frontier, Nature Portfolio, Elsevier, MDPI. His expertise in sequencing data, proteomics, transcriptomics, and metagenomics. Dr. Yogesh is skilled in molecular protein modeling, docking, and simulation. Also proficient in programming like R, Python, SQL, HTML, PHP. So, with the treasure of his knowledge, we are confident that by the end of this session, you will fulfill with the valuable knowledge that will stay with you always. So, without further ado, let me introduce Dr. Yogesh. Hello, Dr. Yogesh.
Yeah.
Before
Hello, thank you
starting the
so much.
session, I just want to yes, just there is a notice for everyone that 100% attendance throughout the session is mandatory for the certifications, and the attendance shall be provided in the last 15 minutes of the session. Thank you. Yes, Dr. Yogesh, you can start. Thank you, sir.
Yeah, thank you so much. Thank you so much for inviting me for this talk. And I'm glad to be here to present my presentation and to share some knowledge about the single cell sequencing and to give you some introduction about single cell sequencing data analysis. So as you know me in that I'm a assistant professor working in the department. Now I'm working in the Department of Data Science, Center of Biomedical Research, Lucknow. This institute is situated in the Lucknow in Sanjay Gandhi Postgraduate Institute of Medical Sciences. So let's start without wasting more time. And okay, so as you know, all of you are maybe aware of these things that this due to the advancement in technology, especially the sequencing technology. Nowadays, a lot of type of technologies of sequencing were uprising day by day. And one of that technology is single cell sequencing. So there was a science report in 2018, and they said that single cell sequencing is a revolution, is just started. So they mentioned that in 2018 that this single cell sequencing revolution has been started and it will go high and high every year. And till date, I think I can say that most of the researchers, most of the scientists, they are now focusing on doing the single cell sequencing. Of course, there are many types of sequencing, but if I will focus only on some RNA-based sequencing, then you may know that we have bulk RNA sequencing, we have miRNA sequencing, we have siRNA sequencing, then this is called a single cell sequencing. So I will introduce you and tell you about what is the differences between the single cell and the bulk RNA sequencing and why single cell sequencing is important these days. Okay, so starting with a little bit brief introduction. So my slide will going to cover this introduction part, and I will tell you what is difference between bulk RNA sequence and single cell sequencing. What is single cell sequencing data analysis? How we analyze the data of single cell? Okay, let's know something more about this single cell sequencing. So as I told you in an introduction, that next generation sequencing. Now the present era is of next generation sequencing. Let's start with the very basic thing. Let's understand what is the sequencing. So sequencing in the context of genetic and let me turn my pointer. Okay. Okay, great. Okay. Okay. So sequencing is in the context of genetic and annotation genomics, refer to the process of determining the order of nucleotide, where you see the adenine, thymine, cytosines, and guanine in DNA form or in the RNA molecule form. Okay. So we have two type of like genetic material within the body, which is one is DNA and one is RNA. You must know, you may be knowing about these two things. So the next is the introduction to next generation sequencing. Then let's understand what is sequencing actually. So RNA sequencing. So RNA sequencing, it is a specialized technique for determining the sequence of nucleotide in RNA molecule, and it is particularly valuable for understanding gene expression pattern and can provide insight into how genes are regulated and which genes are active in a specific biological sample. So it also helps in identifying and diagnostic biomarker for initial experiment. So this is a very basic workflow of sequencing, how we do the sequencing actually when we get the sample, no matter if that sample is of human sample or a mice sample. So the first step is the sample preparation. Sorry. So first step will be the sample preparation. Then once your sample will be prepared, the next step will be the library preparation. If it's a RNA sample, then you have to prepare the RNA library. And then third step will be the sequencing method. Then you have to give your samples after preparation of library to the sequencer machine. Then with the sequencer machine, you will do the sequencing. And the last process, the fourth step, is bioinformatics analysis. So after doing the sequencing, the lot of data will be generated in the form of raw data format, which is called like FASTQ format. So then you will get these FASTQ files, and you have to do on the fourth step, then you have to do the bioinformatics analysis. So this is the process of doing the bioinformatics analysis. At the end, you will get the raw data in the FASTQ format, BAM format, and VCF format. So it depends upon what type of data you get from the sequencer machine. Okay. Yes, there is a one question. I don't know. Yeah, someone raised their hand, but let's continue without wasting more time. Then I will take all of the questions when there will be a session of hands-on session of single cell sequencing analysis. Okay. So let's understand what is the concept of bulk RNA and what is the concept of single cell RNA sequencing. So you must know. So big question comes in our mind that why we need single cell sequencing when we are doing the bulk RNA sequencing. So what is the need of doing the single cell sequencing now? So the good example all of you may be known or have like this daily example in your life of making a smoothie. So smoothie is something like a juice, or so in this smoothie, if you are making a smoothie of different fruits, so for example, you take pineapple, you take cherry, you take berry, you take apple, you take orange, and you make a smoothie. So that smoothie contains a different fruits, you know. So that smoothie contain different fruits. So let's consider these individual fruit as a different cells. So now what you are doing, you are mixing every cells and making a smoothie. So that is the concept of bulk RNA sequence. So you are mixing all the cells and looking how these genes are showing the expression in its combination form in a body in particular condition. Suppose, for example, if you take the sample from the normal patient, if you take the sample from the disease patient, for example, the patient from the cancer tissue of that patient. So you do the bulk RNA sequencing of the normal patient. Then from the tumor sample, you also do the bulk RNA sequencing. And what you will get the average expression of gene in the particular tissue sample type. Okay, you will get the result in the form of average expression of gene. But if you will do the single cell sequencing, you will get that what is the level of expression of particular gene in which cell, because now we are not making a smoothie, we are looking the expression of particular genes in different condition of individual cells. Okay. So for example, if you are sequencing banana, for example, banana is the one cell, then cherry is the one cell, then pineapple is the one cell, and you will look the expression of genes, how much that gene is expressing in particular cell. So this is the difference between the single cell and bulk RNA sequencing, for example. So what organism you can sequence? You can also sequence human, mice, plants, anything. So let's understand with this example the difference between the single cell and bulk RNA. So bulk RNA is it give you the result only the average expression of all the genes. So you can see these are the all cells and these are the cells. These different color shapes are considered as cells. So and these are the genes which are showing the expression in combination, right? And on the other hand, on the right side, you see the single cell next generation sequencing. So you will see that these particular genes, they are associated with the particular cells and showing the expression within that cells. You can see the separation. You can easily see the difference between these two. Okay. So with the bulk analysis, gene expression is average across the cells, and single cell sequencing shows the gene expression by individual cells, providing a much deeper and more specific view of cell-to-cell variation. Sorry, I don't know what happened to this. Sorry for this disturbance. Okay, so now let's understand this example more deeply. Single cell RNA sequence relieves cellular heterogeneity that is masked by bulk RNA sequencing method. For example, this is a tissue. So you took a sample from like any tissue. For example, you are taking this sample from the cancer tissue. So you see the different color is representing a different cells here. Okay. So what you are doing on the downside, you are doing the bulk RNA sequencing, and on the upside, you are doing the single cell sequencing. So for take an example of this bulk RNA sequencing. So you took all the cells, you put all the cells within the pipette in this tube, and then you perform the sequencing. And what you get after doing the sequencing, you get the average gene expression from all the cells. So these each color represent, for example, this each color represent the cells, and you are what the results each you are getting the average expression of each genes. Okay. So there are number of genes. Sorry, each color represents the gene, and this is a single like a column you get here. And now we cannot see what is the cellular heterogeneity when you do the bulk RNA sequencing. You can only see the one thing that which gene is showing high expression, which gene is showing low expression in particular condition. Suppose if you are comparing normal tissue and tumor tissue, you can only see, okay, this gene showing high expression in normal condition, this gene showing high expression in or low expression in tumor tissue condition or tumor condition. On the other hand, if you will see the single cell sequence analysis, so once you take the tissue, then you perform the single cell. So at the time of sample preparation, what you are separating, you are separating the each cell separate by using some technique and machines like 10x machine. So there are there are different machines by which you can load these cells and you can separate these cells. After doing this, then you have to do the sequencing. Once you do the sequencing, you can see example here, each cell type has a distinct expression profile. So this particular cell type had these genes showing high or low, whatever. Then these cell type have these genes showing high and low. And for similarly, these each cells have different genes showing the different expression. And when you visualize them, what you can see the separate different clusters, which reveals the heterogeneity and subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the expression. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpopulation expression variability of 1000s of cells. So here you see, okay, these are the cell type. This is the variability. This is the subpop maybe T cells, this is maybe B cells, this is macrophage, this is dendritic cells. So now doing the single cell sequencing, you can easily separate the cell type. And within these cell type, you can also see that which gene is showing high expression in which cell type. So this is the like basic concept of doing the single cell sequencing and understanding the difference between the single cell and bulk RNA sequencing. Okay. So now let's understand what is a little bit insight we can get from the single cell sequencing, what we can learn from the single cell sequencing. So for example, as I told you, we have healthy tissue and some pathological tissue. It could be anything, pathological condition, which is not normal tissue. It's considered as a pathological tissue. So we took the samples. And then we expression profile. We do the single cell data profiling. After doing this profiling, then we are performing the single cell analysis by using single cell modules and single cell analysis packages. So what you see the difference between these two cells. So for example, when you took the sample from pathological condition and when you took the sample from healthy condition, so you see the differences between these. So for if you will focus on the yellow color, though, so in the yellow color you see the yellow balls, they are a little bit scattered, they are means disturbed when there is a when we took the sample from the pathological condition, they are not tightly clustered together. And these cells, they are more tightly clustered together compared to this one in the compared to the pathological condition. Then you see the green part, of course, they are not tightly, so they are not, because they are not normal, so they are more disturbed compared to the healthy condition. And one thing you see that there is one additional color circle, which is red, which was not present in the normal condition. So we can conclude that there is a one cell which is more active when there is a disease in the same tissue. So these cells which is more active and which come up as compared to healthy condition, we can consider, okay, this cell might be responsible for this pathological condition. So this is a very general example I gave you how these comparison works. So what type of analysis you can do with this single cell sequencing? So within what you can do actually, you can stochastic variability of transcription. You can see the stochastic variability of transcription. You can also learn the regulatory network inference. You can also learn allelic expression pattern, scaling law of transcription, and this is the within the cell type. And when you compare these two cell type, you will see and you will learn the biomarker. You can identify the biomarker. You can see the post-translational differences between these two conditions. And between the tissue, if you compare between the tissue, healthy and pathological, what you can learn the cell type composition. Here you have 3 cells. Here you have 4 cells. So altered transcription in match cell type you can also see. But there are a lot of other things what you can learn from doing the single cell sequencing between the two condition, healthy and pathological condition. So you have to do more practice to learn more. Okay, let's move further. And okay, this is just an example of preparing the data set, how you do the single cell sequence. Of course, I told you this before. You do the tissue preparation, then use single cell isolation. You have to do the single cell isolation. There are methods by using those methods, you have to do the single cell isolation. Then after doing this isolation and library preparation, you have to do the sequencing of the single cell data samples. Then after doing the sequencing, you have to do the single cell data analysis by using the different programming packages which were run in the R or in the Python. Okay, so going a little bit quickly in this because there is a very deep concept and it will take more time. I will tell you how this concept actually works. So, for example, you have some beads. Those are the magnetic bead which attach each cell to itself. So, for example, you can see in this tube, you have different colors beads. These are the 10x barcode gel beads. So these barcode gel beads have a small sequence attached with them. So this sequence is a very unique sequence which was attached with these cells. So once these beads are now pass through this chromium next gem chip, so you will see these beads get labeled. So you label these cells. So every on the cells we are attaching on this bead, we are attaching a cell enzymes. Over here you can see the small circle over there. Then after the attachment we create a gems. This is an oil emulsion, or you can say it's a small oil drops. Inside that oil drops, these small beads were captured. And after this, what actually you get? So after doing this process, what you see in this oil actually, so you have this like small sequence, chunk of sequence, where there will be a this region, this black region called true sequence read, which is from the tissue. Then we have barcode, this green one, which we attach with this sequence. Then we have a red one, which is called unified molecular identifier, which is a very unique molecular identifier. Then we have a mixture of these, and then we do the cell lysis of these, and we replicate these beads. And when you see here, you have read UMI, and then we attach the poly a tail with this, and then we perform the sequencing. And you see the template switch oligo primer. We are doing the synthesis of these sequencing. We create the single cell complementary DNA from this polyadenyl mRNA, and then we are converting these into the DNA from cell surface protein and feature barcode, and then we are doing the transcription extension of these, and in the final step we get it the cDNA library. After this, we do the pool cDNA amplification. We amplify all these preparation, and after amplification, we do this. Okay, if we want to attach, we also do one more thing over here. We can also, it's actually optional. Some people do this, some don't do, and it's totally based on the requirement of the research question. So what is that? We can also attach the VDJ or antibody tag with these sequencing at the time of sequencing, which these antibody are specific, which tell you that, okay, this particular gene is going to produce this particular protein, okay, within the cells. So, okay, let's move forward. After doing this preparation, what you get is the single cell sequence library. Then what you can also sequence using the single cell sequence is the CiteSeq, which is CiteSeq is known as a cellular indexing or transcriptome or epitome by sequencing, which is also called ADT, what I just told you, antibody derived tags. So these are the small tags, and this is a method for performing RNA sequencing. Information on surface protein. So if you will also sequence this ADT with the single cell with a single cell sequence, then you will also get these attached protein which are attached with the particular cell or on the single cell level. So how you do this sequencing of these ADT is you have a structure of an antibody derived tags. So this is the structure of antibody. Then you have some PCR handles. Then you attach this antibody barcode with this sequence, and this is an example of a sequence now. So this looks like this one. So you bind the ADT to the cell of your interest. So this is antibody, and you are binding this antibody with the cells. Then once you bind these antibody cells with these cells, then what you perform a single cell droplet capsulation. Then you go and pass these to the 10x machine, and this 10x machine do its functioning. and they trap these all antibody tagged in a single droplet. And these droplet microbeads you can see here. You will see here the this antibody. You will see the sequences and the microbead. This is a magnetic microbead which will attach with this within this droplet. Then you do the cell lysis in the droplet microbead. You will see the lysis was going on, and here you have these magnetic beads. Then you do the mRNA and antibody oligo hybridization to RT oligo. And after this, what you do, you will do the sequencing of greater than 300 base pair. You prepare the library, then you perform the sequencing amplification, and in the end, you will get this pulling of library and sequencing. Then you perform the sequencing with a sequencer. So this look like very complicated, this concept, but this concept only add few things. The first step, which is very different from the bulk RNA sequence, is this: the tagging of the antibody with the sequence. Then this machine, which we use the different machine, what we don't use in doing the doing the bulk RNA sequencing. So we have a different machines actually for doing the single cell sequencing. So those machines are called 10x machine. We have Rhapsody machine. So they have like different concept. They have different method and different technology. So that's why this looks like little bit complicated. But the difference is here this tagging. Difference is here how these cells were trapped in a single droplet beads. We attach the magnetic bead with them. Then the amplification process is similar what we do in the bulk sequencing. And then we have the sequencers. Sequencers are okay. Then let's move further. So next, what we can do VDJ sequencing as well, which is called variable diversity joining T cell receptor. Actually, what are these VDJ? These refers to the process by which T cells, especially this VDJ sequencing was done when you want to study the T cells only. You cannot do the VDJ with the B cells. Of course, you can do the VDJ with B cells, but they will call the BCR, not TCR. Okay, but you cannot do the VGA with other things, like you cannot do the VDA with macrophage or with dendritic cells or endothelial or epithelial cells. So let's see. These TCR refer to the process by which T cells create unique receptors that are able to recognize specific antigen in single cell analysis. So researcher can study the diversity of T cells receptor at the single cell level, which allow for a more comprehensive understanding of the immune response and the role of T cells in various diseases. So these T cells or doing the T cell sequencing is very important over here. So let's see what's the algorithm of this T cell sequencing. So you have this tube, you have all your like samples in this tube, and you have this VDJ library in this tube. Then you read this library, you assemble the contigs, then you annotate these contigs, and then call cells. Then you get, after your annotation, you will get this variable, diversity, and joining region of these cells. So you have cell 1, 2, 3, then you generate the clonotype of these cells, and each clonotype then each clonotype are the unique clonotype. They generate the unique sequence actually. And then these clonotype also attach or complement with the particular cell, single cell type. Okay, then let's move. So what are the challenges doing the single cell sequencing here? So first of all, the first big challenge is the quality control at every step of the process. So you have to be very uh, you have to be an expert in doing the single cell sequencing if you want the better quality of sample, if you know the better quality of results, or then you have to be a more expertise in doing the single cell sequencing. You have to be more expertise in handling the whole process. If you do some mistake in any part of the doing this process, then you will lose the cells or you will contaminate the cells, because it's very like complicated or sensitive method of doing this single cell sequencing. So it's also possible that you will get rare population might not appear in the initial sample quality if it's not sufficient. And what you yield, the yielding is also another challenge of the single cell sequencing. So recovery of 50% claim by every manufacturer or every sequencing machine or sequencer machine, manufacturer, they claim that we do the recovery of 50%. If, for example, if you are loading 100 cells, this is 100 cell is a very short example, just for an example. Otherwise, the minimum, like if you are loading 10,000 cells, so there will be a chance that you will be only recover 5,000 cells. And of course, doing the sequencing is also a very costly. So it's a very average amount. I'm telling you, per sample, you will cost 1,000 euros, which will be around I think 80 to 90,000 Indian rupees. So what we can do, we can improve the yield and recovery. We should do these things and we have to follow these points. So follow the manufacturer recommendation, what they recommend, because every manufacturer, every machine have like different protocol and different precaution what you have to take. Use high quality material, what they provide you. Use recommended equipments. Minimize pipetting errors. Of course, I told you that you need to be expert if you are handling the single cell sequence. And at the end, when you get the sequencing, then you have to do the single cell sequencing analysis, which is the main focus of this presentation today. So after you get the single cell data, I will also tell you in the hands-on training what type of data you will get after doing the single cell sequencing, what type of data you get directly from the machine. And this picture, you see that this colorful picture, this is the like very end product of doing the analysis. So after you do the step-by-step analysis, and at the end of this analysis, you get this like very beautiful picture. But this beautiful picture have a lot of information in this. So, okay. So each color cluster is representing the each cell type here, which you will learn in next 1 hour. So these are the very standard method for doing the single cell sequence analysis. First one is standard pre-processing workflow. Second one is QC and selecting cells for further analysis. Removing the low quality cell or empty droplets will often have very few genes. I told you that we have to after in the droplet all these like material capture in the droplet, and sometimes it also happened that instead of one cell, the two cells were captured in a droplet, and those two cells get into the sequencer and sequencer sequencing two eyes. Okay, so we have to remove those duplex or doublets when we do the sequence analysis. So cell doublets or multiplex may exhibit an aberrantly high gene count. So low quality dying cells we have to remove if there will be a sequence, if there will be some error while preparing a sample and your cells got died, or maybe your cells contain some mitochondrial genes, or because of maybe it could be any reason. It could be any reason with any reason, maybe the transportation reason. You prepare the sample, you send the sample to the sequencer, and the sample was not handled properly, or it was not kept at the appropriate temperature, then your cells will die, and then you also get some mitochondrial contamination. So you have to keep these all things in your mind when you do or dying cells from exhibit an extensive mitochondrial contamination. Another step is, once you do these two steps, then you have to do the normalization of the data. The next step is identification of highly variable features. So variable feature here, I mean the genes. Feature is gene. So you have to identify the highly variable genes. Then you need to do the scaling of the data. Then you have to perform linear dimension reduction, because you have seen that when you do the bulk RNA sequencing, you got sequence 20,000 or 30,000 of genes. And if you have seen this matrix, bulk RNA sequencing matrix, so you have these things like you have gene name in the column, and or maybe the vice versa, you have gene name in the row and sample in the column. So okay, so if you have bulk RNA sequence, it's quite easy to fit that in 2 dimension. If you have 2 samples, you can fit it in 2 dimension. If you have 3 samples, you can fit it in 3 dimension when there is a case of bulk RNA sequence. But in the single cell sequencing, you sequence 10,000 cells with a 25,000 genes. So it's difficult to fit all these like orientation within a single dimension or 2 dimension. So that's why we have to do the linear dimension reduction, where you want to fit all these dimensions to visualize on a single map or a single plot. So then you have a cluster. You have to do the clustering of cells. So for clustering of cells, you can run either UMAP or TCNA. These are the statistical logical method for visualizing the high dimensional data on a single plot. And at the end, you have to find the differential gene expression. So these are the several packages which you can use for doing the single cell sequence analysis. And these packages are Seurat, which we are going to use today. And this runs in the R. If anybody of you are familiar with R, I suggest you to download this Seurat. Just type the Seurat in your CRAN from your RStudio and download it, or install this Seurat package. Then you can also follow me when we will perform this hands-on training at the time of hands-on training. And if you don't have laptop, or if you don't aware with this R programming language, or if you are not able to install this Seurat package, or if it take more time, then you just sit, relax, and see what I'm doing and follow me. Okay, these are the under next visualization features what you will get. I think I will teach you all these graphs, how they look, why we plot these graphs in the hands-on. Okay, I will teach you step by step what these graphs look like. So this is an example just for the QC. When you run the QC, you will see these type of things, these type of beautiful graphs. What they mean, I will tell you in the hands-on. Then you will do the normalization of data and find the variable features, variable genes. Then you will perform the linear dimension reduction. You will perform several statistical method. This is an elbow algorithm to find out, okay, how many cluster we can get. Then after all, at the end, you will plot this beautiful graph where you will see that each cell type were clustered together. And after doing this, then you will do the annotation. We will do the annotation of these cell type. For example, we did the annotation that this blue cluster representing the B cells, this representing the activated CD8 T cells, this shows dendritic cells, this shows CD14 monocyte, this is natural killer cells, this is naive CD8 T cells, and this is CD4 T cells. So like this, you will do the annotation of this cell type. So actually, these are the basic steps. For examples, I will tell you in hands-on. These are the just representation how we came to know that which cluster is which cell type. This will come in the hands-on training. Then there is some extensive analysis change of particular cell in the time period. For example, if you see that I have annotated these different cluster with a different cell type, and here you will see the row arrows going right, left, and connecting with each other. So these arrows are representing that there is a transcriptional change between these cells. For example, if I say that this, for example, this TH1 cell is now converting to the TR1, and this cell is converting to the other cells, and this cell is converting to other cells. So this comes uh, under the category of trajectory analysis, which is the extensive form of doing the single cell analysis. So after doing the very simple analysis, you can perform like trajectory analysis, you can perform interactome analysis, and you can perform velocity analysis and so on. So there are more further extended analysis what you can do using the single cell, and those each and every analysis is depend upon your research question. Okay, this is an overall workflow. And now I would like to end up my presentation here. Then I will try to give you the hands-on as I can. So it's I think uh 10 minutes. So we have a 10 minutes. We can take a question in this 10 minutes, and then we will move to the hands-on training session. Is it okay? So I can see in the chat box we have 99 plus questions already. What happened upon the attendance? Okay, these are the logic, not it will be shared. Okay, sure, agenda because of okay, so all the questions are related to the omics, Dr. Omics. Hello, hello.
Yes, sir, you are audible.
Yeah. So I think all the questions I can see in the chat box are related to you, right?
Yes, sir.
Yeah, there is no technical question here. So let's move to the hands-on session, I think. Okay. I hope you must be having the R Studio in your systems. If you don't have, then it's okay. But if you have, then it's good. Okay. So, so can you see the screen or not? Okay, I'll share my screen again. Okay, let's share the screen. Okay, so I think everyone can see my screen.
Yes, sir.
Okay, great, great. We can hear you. Okay, great.
Sir, before starting, there are 3 to 4 questions from
Yeah,
the
I'm looking like yeah, I'm looking the questions. Okay, there is one question for doublet. Okay, I will reply. So there is a question, I will read it. How to separate the data which comes from cell doublets or multiplex in single cell sequencing. So there is a very nice package called Doublet Finder. So what you have to, you can use the Doublet Finder package for removing the doublets. And the catch in that package is that you have to you have to do some hit and trial to set up the cutoff range in the doublet finder. So you can start from the 5% that you can give it a like a percentage number that we want to exclude the 5% doublet. We want to exclude the 10% doublet from the samples what we get. So the tool name is Doublet Finder. I will write it. Doublet Finder. Okay, then there is another question. Uh, Okay, so as you see from the name is the unique molecular identifier. So yes, there will be a unique UMI. Then the another question is, what is the average read length in next generation sequencing? Okay, generally average read length depend upon the type of machine you are using. Maybe 100, maybe maybe 1,000. It depends upon what type of sequencer machine you are using. Then, so we need to create new project in. Yes, you need to, if you want to start it and you want to follow me, you have to create a new project in R, and then you can follow my command. You can type what I am typing, okay? Or you can just look and type the command, what you see in my screen, on my screen, okay? So can we study disease of plant with single cell? Of course, we can study the disease of plant, but I told you that this single cell sequencing is very costly method. And of course, we can study anything which, any organism which have DNA, RNA. Yeah. Need a summary of these topics. Okay, we will give you. Yes, we can see now. How can we do single cell sequencing abiotic stress plant? Actually, I don't have experience of doing the single cell sequencing in plants, but I think we can do this. But you have to explore, you have to look some publication if there is some publication related to your question, abiotic stress plant. Yeah. Then should we perform these analysis in R in Linux environment? Yes, you can perform this in Linux environment. Of course, I choose Windows because I like Windows and it's very convenient, and you can also do it in Mac as well. So it's not dependent, it's not necessary that you have to do this in Windows. You can choose your platform, whatever you like, Linux, Windows, or Mac. Language, if you want to, if you like R, you can do it in R. If you like Python, you can do it in Python, but packages will be different. In R, the package will be the suite, and in Python, package will be ScanPy. Of course, there are more packages, but these are the two packages which are widely used for doing the single cell sequence analysis. Then, yes, this question repeats. Can we use the rigidity of clustering different along without the involvement of new cells population upon comparing healthy and pathological sample as early diagnostic? Yes, of course, you can do. Actually, this single cell analysis is the best way to identify the early diagnosis biomarker. So we can perfectly identify the early diagnostic biomarker when you do the single cell sequencing. And precisely you can tell that this particular gene which is associated to macrophage, which is associated to T cells, could be the diagnostic biomarker. So this additional thing you can identify using single cell sequencing. So another question is single cell better than ADT in terms of the data analysis? No, actually ADT is a part of the single cell. So when you do the single cell sequencing, you have to tag those sequencing with the ADT, antibody derived tags. So those antibody derived tag will confirm you that this particular gene going to code that particular protein. Okay. This is the integrated part. ADT is the integrated part of single cell sequencing. Like you can say the add-on part. So how do we identify biomarker and check allelic expression pattern and post-translational differences? That could be checked when you perform the additional analysis of the single cell data. You can also identify the post-translational transcription factor. There are another tools for doing this on the single cell data. So that is Scannic, I think. Scannic, yes, which run in Python. And Of course, if you want to see the trajectory of these particular cells, then you perform this trajectory analysis. And if you want to if you want more deeper information on this, then you have to perform the velocity analysis. Please mention filter from where we can get publicly available data for. You can easily get this single cell data from SRA database. So if you will go to the NCBI site, just select the database SRA database, and in that database, you will see the raw data which everyone has submitted. The researcher have submitted this single cell data in this SRA database, but you have to find out. Actually, you have to find out because every raw data, no matter if it's single cell, no matter if it's bulk RNA, you have to, and when you have to do the publication, you have to submit the raw data in SRA database. So you have to little bit dig into these databases. Okay, which one is for single cell, which one is for bulk RNA. And the very simple way for finding the single cell RNA data is that go to the publication, go to the section data available, click there. There will be an ID, SRA ID. Click on the ID and you will get directly that. You will be jumped on this SRA database and you can download the data. Sir, for performing now we will need single cell RNA data. Will you provide it as? Yes, of course, I will tell you from where you can get this data. So I will tell you, okay, let's okay, everyone, let's if you are able to do, you can open this. Okay, can you see my Google browser?
Yes, sir.
Okay, just type and follow me. Just type Suret and go here. And perform this example. And click here on the raw data. And you will get this raw data. Open your raw data. So I already untarred this raw data. So you can just do it again. So you have to unzip this. Then you can change the name or whatever. If you don't want to change the name, you can keep it same. But I suggest you to change the name. Demo. Then you can find this data type inside. This is an example data barcode. You will find the 3 file barcode, genes, and matrix files. Okay. And then you have to follow me. Every time we perform single cell, we need to be incorporated or we can include. Yes, it depends upon if you are doing the JAK sequencing, means simply the single cell, or we are including the ADT. We are adding this ADT at the time of preparation of sample, and we are sequencing the ADT with a single cell. It depends upon. We can do it separately or we can also do it combinedly. So question is that every time we perform single cell RNA data, ADT need to be incorporated or we can exclude based on our aim of the experiment. We can exclude. It depends upon your choice. But if you will include, then you can reduce the cost of sequencing. So that's why mostly people who need to do the ADT, they add this ADT with the single cell sequencing. Yes, you need to download the Suret and R Studio with the R. Then why do we use RNA for single cell data analysis and not DNA? No, we don't do we don't do the DNA. So we only use the RNA sequencing. DNA very basics. How to use R system for analyzing. Okay, I'm going to teach this. So we can get duplicates, especially in history. No, I don't, I never said that we can treat cancerous patient. We can only analyze the data of cancer patient. And once we are confirmed, then actually you cannot directly do the experiment on patient. You cannot start treating the patient just with the analysis. Okay, next question is. Okay. There are no more questions. Then let's proceed with the experimental part. So now let's do the analysis part. I hope some of you have downloaded the sample data set. And if not, then just sit back, relax, and follow me. So let's begin with this analysis. So I have previously written this code in the full form. But if you are new, so I will also follow with you. So you can write a new project or you can just write a new file in the Markdown and give it a title like demo. I prefer to save it in HTML and just follow me. Okay, I will save it and I will save it in the name of demo as demo. Okay. Okay, just delete all the things. We don't need these things. Now, the first thing you have to do when you are very fresh and new, here is you need to install the R package. So what you can do, you can just either you can type command install.packages Suret and run it. I have already installed this package, so I will not install it again because otherwise it will change my version. So what you can do here is, sorry, either you can do this or not, it doesn't matter. Or what you can do, just copy this because now Suret package is available in the CRAN. Go to the tools, install packages. It's slow. Just type here Suret and it will come up automatically and install it and wait. I know if you are new, then this installation can take 5 minutes to 10 minutes, depend upon speed of your internet and your computer. It can also take more time. So it's better to follow me and my instruction, what I am doing here. Okay. So once you will be installed with all the packages of Suret, then what you have to do is the first step. You know that after doing any installation or if you want to run any package in R, you need to call the library of that package. So this is a very general library, dplyr, for doing some statistical analysis. I'm calling this package dplyr. Then I'm calling the Suret package over here. So now I'm loading the Suret package. This package is for doing the single cell analysis, all the analysis. Okay. So let's go to the console. So now I successfully load this package. You can see it's loaded here. Warning, Suret package was built in under R version, this version. Now you can also load some other additional library. This is called Patchwork, which is just for doing some basic statistical. It's final step is, if you remember, we have downloaded the basic this example file over just now. So that example file contains matrix file, barcode file, and feature file, means the gene files. So now I'm loading using this command, pbmc.data read 10x. Why 10x? Because the file what we have downloaded just now, it was sequenced by a 10x machine. That's why we got three different files: matrix file, barcode file, and gene or feature file. Otherwise, if we change the machine, sequence the machine, like if we sequence this single cell sequencing data, single cell sequence with the Rhapsody, for example, Rhapsody is another company or another machine for this doing the single cell sequencing. So if it was done by Rhapsody, then you will get only the matrix uh, in the form of, like, in the column of the matrix, Excel matrix, you will get this gene name, and in the rows of the matrix, you will get the cell type, means the cells, and the cells code will be in the form of sequence. Okay, it will be the short, very short, like 15 to 20 nucleotide. So you will get this in the form of matrix. But here, because we do the sequencing with this 10x, so we get these three files, and these three files are connecting with each other. So you have to load all the three files, what you see here. So I give the path. For example, I give the path here, practice folder. Then we have this folder. In this folder, we have the human genes, human data, which was sequenced. Then we have these three barcodes. So what you have to give, you have to write path only until HG19 or whatever it will be. It depends upon your folder name. So you have to write on, you have to get path only up to here, and then you will load the data. So now I'm going to load this data, and I will also show you how this data looks like. So let's see after this how this data looks like. So I think this data will be a very huge matrix. See, so this is very huge matrix data where you will see there are genes, name of genes, and in the column, these are the sequences, or these are the different cells. Each this column, and these are the name of the genes. And each dot is considering as a zero or a blank over here, not only the blank, but zero. And when there will be a expression value of this gene in particular cell, then you will see the value over here. Okay, this is the type of matrix what you directly get. But in the 10x machine, you get the three different files. And these files are matrix file, barcode file, and feature file. Okay, after loading this example data, now we have to create the Surrogate object. So we use this function, create Surrogate object. Surrogate is the package what you are using. What it will do, it will count the matrix pbmc.data, and it will make a project with the name pbmc3k. You can also change the project name of this project. And now we will include the minimum number of cells. These cells, we will take the minimum number of cells, 3, to create the Surrogate object, and minimum number of features, means minimum genes, we are taking 200. These are the very default value for these minimum number of cells, and taking minimum number of features or minimum number of genes. So now, using this default value, we will run this command. And after running this command, we created the Surrogate object here, and it did some changes according to himself. It's just where he find the underscore in the gene name, he's replacing with a dash. It doesn't matter. You don't have to take care of this. So now, after creating the Surrogate object, let's see how this Surrogate object looks like. So now, this is the whole statistics of Surrogate object. If you read this statistics, so now we have loaded 13,714 genes, means features. Across, we have 2,700 samples, means cells. So in 2,700 cells, we have 13,714 genes which are expressing in these cells. Okay, this is the meaning of this. So now, as I told you that there are some few steps for doing this analysis, still we have only loaded the data. We haven't followed the other step, but the first step, if you remember, is the QC step. Okay, so and standard pre-processing step. Standard pre-processing step. So let's do the standard pre-processing step. The step, I have written something over here. So the step below compass the standard pre-processing workflow of the single cell sequencing. As I told you in my presentation that what we have to do in these steps, we need to check the number of unique genes. How many unique genes are there? We need to see the low quality cells or empty droplets if there are some. We need to check the cell doublets, and similarly the total number of molecules detected within a cells. How many molecules are there means how many these cells are there and how many genes are there. We have to see the percentage of read that map in the mitochondrial gene. We have to see the mitochondrial gene, and then we have to remove those mitochondrial genes. We will check the low or dying cells or the mitochondrial cells, and there are different step. There is a one step for removing the mitochondrial genes. So, for example, if you see below, if you see the name of the genes, they are in the row, and the cells are in the column. So now we want to see how many mitochondrial genes are present in how many cells. So let's do it. So you have to keep one more thing in your mind, that when you are dealing with the human data, the gene name will be all the letter of the gene name will be in the capital. And when you are dealing with the mice data, all the gene name except the first letter, all the letter of the gene except the first letter will be in the small letter, in the small cases. Okay. So that's why let's find out the empty. Empty means the mitochondrial gene. So every gene which is of mitochondria, they should contain this empty and dash in it. So now we are grabbing all the genes which contain this empty two words or two alphabets. Let's grab them. So now I grab all the genes which contain empty, M and T. And this specific pattern will be identified only in the mitochondrial gene. Okay, remember this. You must be also thinking that if this type of pattern will come in other gene, then what happened? But I'll make you sure that this type of pattern of this regular expression will not be found in other gene. This is specifically will be found in the mitochondrial gene. Okay, now let's visualize how it looks like. This pseudot object, how it's look like. So now pseudot object look like this. So now we have in the row, we have these cells. And if you remember, we made a name of the project PBMC3K. Then we have number of count of RNA. So we have these RNAs in particular cells, and then we have features, these 779. These are the genes in particular cells. And then we have this percentage of mitochondria in each cells. So now let's remove these mitochondrial genes. So for removing these mitochondrial genes, how we will come to know that how we will remove these genes. So for removing these genes, we have to first visualize it, how these genes are looking and in what percentage these mitochondrial genes are present in our data set. So for visualizing, we will use this another function, which is called VLN plot, which is PB. Now we are using the VLN plot, and here we have this PBMC, which is the pseudot object we created above. Then we will use this feature function. Feature means the gene function. And here we are considering these feature as we are number of RNA, number of count of RNA, and the percentage of mitochondria within this sample. So let's see. And now we are making a 3 column. So let's run this command. And on the right side, you will see, okay, not on the right side, I make it here. So you will see the picture, what I have also shown you in my presentation. This first column is showing the number of genes per RNA in RNA, all the genes. Then you have the number of counts in RNA. Then you have percentage of mitochondrial gene. So here you can see the percentage of mitochondrial gene is not too high here. So this means that while processing this sample, the person who processed this sample or who did the single cell sequencing of the sample was well trained, and he's a perfect example of number of mitochondrial gene with this. So there is no chance of contamination. And by default, we have considered the acceptable percentage of mitochondrial genes for doing the single cell analysis further is 5%. And it depends upon the sample type. And what I means acceptable percentage, I will tell you in the next step. So let's visualize these things in different form. So for visualizing these things in a different form, we can also visualize in the scatter plot these two things, number of count and number of mitochondrial gene, and we can correlate these number of counts and number of features with each other. So let's draw it. So I am drawing plot 1, then plot 2, and now I combine these plot 1 plus plot 2. So I'm combining these two plots. Okay. So I'm using the shortcut key of keyboard for running these commands. So you can also run, like you can click here and just click here, run selected line, and this command will run. Okay. So I'm using my shortcut key from the keyboard to run these command. So now you can see on the x-axis you have number of counts of RNA, and on the y-axis you have mitochondrial percentage gene, percent of mitochondrial gene. Then in the right side you have number of count and you have number of features. So you can see that these mitochondrial genes, they are very less, above 5%. So now the default cutoff for this type of data of like excluding the mitochondrial gene is 5%. So what we will consider here, we will consider all the cells which are having less than 5% of mitochondrial gene. We will take those cells as a good cells. And if the cells having more than 5% of mitochondrial genes, we will exclude those cells in this command. Okay, so what we are doing now, we are subsetting all these functions you see here in the Seurat object. These functions either are made by the package owner or the developer of the Seurat, or some of the functions are the by default function of our language. So this is a subset. So we are subsetting here. So we are excluding. So what we are doing, we are selecting the number of features. So let's select the number of genes. So we will see that take all the genes, all the cells which are having greater than 200, all the cells which are having greater than 200 genes, and all the genes less than 2500. And how we consider this less than 2500? Let's come to this graph. So what we have plotted here, we have plotted the number of features, number of genes. We have plotted the genes here. We have plotted the RNA here. Then we have percentage of mitochondrial genes. So we have to decide the cutoff of number of features from here. So for example, if you will see, if you are aware of the VLN plot, or you can also plot it in some other plot, box plot, whichever plot you like, actually, you can do it according to that. And then you will see here, you have like at 2000, this cutoff looks like good. And above 2000, you will see that a lot of genes are scattered, more scattered. So you can set the cutoff of your genes up to 2000, or you can also go up to 2500 up to here. You only see one scatter gene, but we don't have anything above 2000, so we can take a cutoff up to 2000. Then here I told you above 5% we will exclude these scattered gene because they are not good. So what we are doing here, we are setting this cutoff. We are considering greater than 200 gene should be there per cell, and the total gene should be not more than 2500. But let's do it with the 2000. Okay, let's do some changes. And for mitochondrial gene, we will exclude all genes greater than 500. So now you can also click here if you are using R Markdown to run the command. Okay, now I run the command. So let's see. Let's give you some more example. So if you see the before, we have more genes. So now we have 13,714 genes, okay, and 2,635 cells. And how many cells were before doing this filtration? You can see, let's see how many cells were there. So we have 2,700 cells before doing this filtration. Then we did the filtration and we left with only 2,635 cells. So we exclude around little bit around 70 cells which are having more expression of mitochondrial genes. Okay. So let's go to the next step is the normalization of the data. After removing unwanted cells from the data set, the next step, of course, is the normalization of data. So we do the scaling of this data now. So this is the normalization function of the suite. You do the normalization, and you can also you also have some option. Either you can use these different parameters, and if you don't want to use this parameter, just use this command. I will use this command, very default command. I will not use the parameter of scaling. So I will use the default scaling parameter. Now all the data is scaled. Now whole data is scaled, and how you will come to know that if it's scaled or not. So let's visualize it. Okay, I will type some additional command which were not part of this tutorial, but I will show you. Uh, tu- tu- tu. Okay, then you can go to the RNA, then you can go to the data, scale data. So here you will see the differences. So you have 0, 0 matrix before, and because we didn't scale it yet. Okay, so let's see in the next step how this data will look like. We just normalize this data at the moment. Then the next function will be the find the variable features. So as I have told you in my presentation that we have to find the variable features, means the variable genes which are the most important gene within this data set of single cell. So we have to use this variable feature. We are selecting the number of features is 2000 gene. So let's run it. So now I did the variable feature selection. Now I got the variable genes, which was stored in the student object itself. Now let's visualize the top 10 variable gene here. And if you will see and run this top 10, you will see the name of the genes. These are the top 10 genes which are very active or which are very variable in this data set, whole data set. So these are the top 10 variable genes. So now let's plot and visualize these all the genes. So now I plot and visualize these genes. It's plotting. You will see the plot here. Okay, it's a little bit not too clear, but it's look like. So this is without annotation. This is with annotation. You will see all these genes which I show here. They are plotted on this plot. Okay, so we have non-variable genes 11,714, and we have 2,000 variable genes because we set a cutoff here 2,000 as a variable gene. Okay, now this variable gene will be stored under another subcategory of a variable gene or a variable features within the pseudo object. Okay. So now the another step is scaling of the data. So we need to scale this whole data. So we have option either we can scale the whole genes because or we can scale the only the row of genes. So let's do it scaling. Let's do the scaling. Either you can use this as an additional or you can leave it and it will scale only the 2000 variable gene. If you will not add this all dot genes, it will only scale the genes what you have selected. It will scale only 2000 genes. But want to scale all genes. It depends upon your question actually. If you want to scale all gene, if you want to scale only the variable genes, if you will do this more single cell analysis, you will come to know what's the difference because it's not it will be very difficult to cover everything in this 1 hour hands-on. Okay, so I run this and I scale all the data and the scale data will be stored here. Assay, RNA, and scale data. So you have this scale data, it will be bigger file actually. So now this data is scaled. Okay, you don't see here because it doesn't fit properly in a matrix form when you display it. Okay, let's do it 10. Okay. Okay, it's not the good way actually to visualize, I think. So what you can do, you can see the scale data in your matrix or in your Seurat object where it is stored. You can go to this PBMC. You can see the scale data. It is stored in the form of a matrix like this. Okay, but you don't need to see this. I'm just telling you an example how this scale data looks like when you scale this data. Okay, then let's do one more thing, how we can remove the mitochondrial source of variation. Now let's also scale these mitochondrial genes, these 5% genes. Sorry. Okay, now I'm scaling these 5% mitochondrial genes. Of course, they are also scaled with this, the previous command, but there is no need to run this command again and again. So yeah, you can also skip this. This is just an additional example showing you. Okay. So let it finish. Okay, now it's going to be finished soon. Okay, now it's finished. But you don't have to run this command or repeat this command. So I put it in as command. Now, as I told you that we have a very huge matrix, like thousands, we have around many thousands of genes and many thousands of these cells. So we have 13,000 of genes and 2,000 of cells. So we have to fit those in a single plot to visualize them. So for plotting, we have to run some dimensional reduction algorithm. So first algorithm for dimension reduction, we will run the PCA. So we will use the run PCA function and we will put the student object over here. And we will select all the feature file, means all the RNA, all the genes. We are doing the PCA on all the genes. And just run it. Now PCA function is going to perform. It's running. It will take few time. And running time of each and every function, each and every code of line depends upon your speed of your computer and speed of the data. Okay. Now, after doing the PCA, we created some PCA, PC1, PC2, 3, 4, 5, 6, more maybe, but I displayed only 5 and few PCs. So now let's look which PC or looks better actually. Then let's do the printing of this PC. So I printed PC 1, 2, 3, 4, and 5. So I generated only 5 PC. And now for visualizing this PC, we use this function, viz dimension loading. Okay, sorry. So now we visualize. This is just a basic feature. You don't need to run it again and again, and you don't need to look more deeper into this. So if you see the PC1, it is more like uniform. If you see the PC2, it's less uniform. There is a break in the PC. So you can select, and if you will plot more PC, let's see, you plot 3 PC. So you will see the third PC will be more disturbed, and we have 1, 2, 3 breaks. So it depends upon, it shows that the quality of this dimension will be better in the PC1 and PC2. So now let's display them in a more better way. These PCs, let's display this PC in a more better way. Run this command. So for displaying this PC1 and PC2, we are just plotting the plot between PC1 and PC2. You need the another function called dim plot. This is the part of the Seurat object, this Seurat pipeline. So now we plot a PC1 and PC2, and we can see these dot plots. They are and then if you want to visualize this PC and representation of this PC in a different form, let's visualize this PC in a heat map form, and we are only visualizing 500 cells, okay, in a one dimension. Let's see how it's look. So you can see PC1 looks like this. We have very nice yellow color, which is in a one section. We have blue this purple color in a very one section, and then nice purple color here. So these all genes in 500 cells, they are showing its expression. They are separating properly. So let's visualize in 15 dimension. Means now we are plotting the 15 heat maps. So if you will plot these 15 heat maps, you will see all the heat maps like this. So you have now 15 PC heat maps. So from visualization, what you will consider, which PC you will consider, which looks like better. So from your eye, if you look this heat map, you will see that PC1 is better and PC2 is better because they have separate yellow color from the purple color, and then PC3 is now mixing with each other. So, and the PC4, 5, 6, and every PC are now disturbed and they are mixing with each other. So for further analysis, we should consider PC1 and PC2 dimension. So now let's plot another thing that will decide how many different clusters we can generate from our single cell data. So for that, we need to do the elbow plot. So this is another function called elbow plot. So you can do it elbow plot, and it's by its name, it's an elbow plot. You can see it's an elbow-like. So you consider it is as an arm, your hand, and consider the band in this plot as an elbow. So where you will see the band, we set the cutoff that, okay, let's set the cutoff 10 for doing the, for plotting the clusters. So let's set the 10 cluster actually. So we can say that let's set the 10 for doing the cutoff of this cluster, because we see the band in this plot at the 10. Means you maybe you can also see somewhere else, maybe you will say that, okay, I can see the plot band, elbow of the band at 8, 7, or 6, but it depends upon your choice. But also statistically, it makes sound when you will see the band. I can see, and because I have more experience to on this elbow plot, so I can say that we should consider the 10 as a band of the elbow plot. So we will consider the 10 PCs for plotting these cluster. So now the next step is the find cluster. So find cluster is the another function which will help you to plot that beautiful, colorful plot, what I have shown you. So the main function of this function is to cluster each and every cells in its specific area. So I said that I will set up the cutoff as 10. So I'm setting the dimension cutoff as 10 here. And now I'm doing the find neighbor function. And after running the find neighbor function, I will run the other function, which is called find cluster. Now it will find the number of clusters, and I'm setting the resolution. Actually, you will learn by time that you how to play with this resolution parameter. But at this time I'm setting this resolution parameter as 0.5. So now I run the resolution parameter, and this resolution will give you the number of clusters. Okay, let's identify how this pseudo object now look like. So now, how many clusters we have generated? We have generated 0, 1, 2, 3, 4, 5, 6, 7, 8. Means total 9 cluster. We have generated total 9 cluster by setting the resolution 0.5. If you will increase this resolution, suppose I will increase it 7, then what you will see? What will be the change? I'll show you. If I increase the number of cluster resolution, of course, there is no change. We have this 9 cluster even though. Let's start and repeat these steps again. 0.7, I increase the resolution again. And then let's see the number of cluster. Okay, still we have these number of clusters. Okay, I think this is the maximum value for this number of cells. Let's decrease the resolution, okay? And then let's see what happened. So when you will decrease the resolution, you will decrease the levels, means you will decrease the number of clusters. Okay. Okay, now let's come to the another point. Then I have told you that for visualizing this data and for displaying this data, you need to select either you want to visualize this data in UMAP or you want to visualize this data in TCNE. These are the like different method for just plotting the plot. So now I'm plotting the plot in the UMAP. I will run this function, run UMAP function, which is also the part of something have stopped. Maybe there is something happened to my computer. Okay. So run UMAP is taking time. Okay, something happened to this program, so I will run it manually. Okay. So now I'm running this run UMAP command, and it's showing here. It's running. So it will take time. It depends upon your speed of your computer. Again, I told you. So after running this UMAP, it's every calculation was completed and it's stored in the backend of the student object. And now, after running this, let's run the display command function from the function called dim plot. So I will run over here because something was happened in this. So now I write a dim plot, PBMC, that's my student object, and reduction will be the UMAP because we run the UMAP over here. Now you will run it, and after running, you will see the UMAP on the right side in this plot. If you are running it in the markdown, you will see the plot just on the downside of this. But something happened to this, so I will stop it running. So I will type the commands manually and I will run it side by side. So you can see that nice plot was plotted over here, and each color is showing representing the each cell. And we have 9 cells. You can see here 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 cells. So these cells are the different cell type. I mean, maybe there will be a T cells, B cell, macrophage, dendritic cells, or maybe eosinophil, maybe erythros, or what you can endothelial cells or epithelial cells, gamma delta T cells, and so many cells. There are so many names. But how we will come to know which cell is which cell type? That is our next question over here. So to answer that question, we have to run this function, which is called find cluster. So let's skip these two commands because these two commands are specifically for those if you want to compare the gene expression among the clusters. For example, if you want to compare what are the different genes expressing in this brown cluster and compared to this blue cluster. For example, what are the genes which are showing different expression in these two clusters? But we have to look all the genes which are showing differential gene expression among each of these clusters. So for that, we will run the function called find all marker genes. So this find all marker genes function will run and find the marker genes in each and every cluster. So let's run it. So let's run this function. So now it's calculating the marker genes from each and every cluster. So it's calculating first from the 0 cluster. Then it's called to the 1 cluster, cluster number 1. Then it will go to 2, 3, 4, 5, 6, 7, and 8 at the end. And what we have set here, the average log to fold chain value should be 1. There will be a one fold change in the gene. You can also look, you can type this find all marker and go to the real function and check its parameter, what parameter you can change it. You can change it according to your query, according to your research question, what you actually want to look into that data. You can follow that. It will take a bit time. Every command time depends upon your speed of computer. Again, I will say. That's why this is also sometimes a limitation of doing the single cell analysis. This machine will be also a limitation because this is only a one sample. If you have like 100 sample or 50 sample or 20 sample, 30 samples, which have each sample have 10,000 cells, you can imagine how big your file will be, and your normal computer cannot handle that file. Not even running these commands. So you need at least for analyzing these single cell sequence data. If you have a sample and that sample have a 100,000 means 1 lakh cells, then you basically need the computer configuration should be like 32 GB should be the RAM and 1 TB should be the hard disk, and your PC should be a 8 processor. Okay, Okay, it's done now. So what we get, you have also seen these type of things in the bulk RNA sequencing. Actually, these you get the p-value, you get the average log fold change, you get the gene name, you get the adjusted p-value here. So this table looks like similar to what you have seen in the simply bulk RNA analysis. So this is again the command, just looking for the marker genes between two cell type. I will skip this. So now I'm going to just visualize randomly. So suppose you have these top 10 genes. I got this top 10 genes. So I will copy this gene. Okay, let's plot it. I will type the command VLN plot PBMC. Now I will visualize only these two genes in a feature plot. Okay, so feature plot is the plot for visualizing the gene. So first gene is LDHB and second gene is CCR7. So what these genes means, you have to study these actually. So let's run this and we'll see how these genes were plotted. So this first gene, LDHB, I also don't know what is this gene. So this first gene shows the high expression in cluster 0, then cluster 2, then cluster 4, and of course in cluster 6. And this CCR gene, this gene says this stem cell gene actually, or you can say the naive cell genes. So you can see this shows high expression in cluster 0 and then it's showing high expression in cluster 2. So what I have told you that what's the basic difference between the single cell and bulk RNA is this. In bulk RNA, sample name, you will see, okay, this is the one sample and this gene showing the average expression in this sample. Then there will be another sample and you will see the average expression of this gene in another sample. You cannot see the you cannot see the expression of this particular gene in a cell in single sample. So here we have single sample, but we have different cells. These are the different cells. And within different cells, you are seeing the expression of gene within the cell. So in this cell, it's showing high. In this cell, it's showing low. It's little bit low. And in other cells, it's almost negligible, I can say. So then similarly, you can plot and visualize many other number of genes over here, like I did. I'm not going to plot them. Then let's move on next. So now the step comes how you will annotate and how you will came to know which cell type is this 0, which cell type is this 1, 2, 3, 4, 5, 6, 7, 8. So this question will be answered when you look the gene expression, differentially gene expression table. So here you can see this is a cluster. This is 0 cluster. And in cluster 0, you see 1, 2, 3, 4, 5. These 5 genes, they're showing high expression in 0 cluster. So there are standard biomarkers which are representative of particular cell. If those particular genes, if suppose gene X is showing high expression in particular cluster, so that gene expression X is should have like its unique feature that X gene will going to be show high expression only in particular cell type. For example, CCR7. So CCR7 is a biomarker, or you can say marker gene of only the naive cells. Okay. And for example, CD3 is the marker of only the T cells. And for example, FOXP3 is a marker for regulatory T cells. Okay, so we have a specific marker genes which code a specific cell type. For example, you can see here in cluster 0, if we see the high expression of CCR7 and IL7R, then that will be naive CD4 T cells. If we see the high expression of this CD14 and LYZ gene, then that cluster could be annotated as CD14 plus monocyte. For example, if there will be a high expression of MS4A1 gene, then that cluster will be annotated as B cell. So we have a different marker genes for a different cell type, and those marker genes you can find out from different databases, marker gene databases. Also, you can look those different marker genes in different publications. So let's run the command. Let's annotate the cell type. Okay. So now I'm running this command and I will tell you what this command means. So what this command means. So now I'm assigning these all names in a very blank like row, which is called new.cluster.ids. So I'm creating a one column, sorry, I'm creating a one column which is a blank, and now I'm assigning this column a name. So I'm assigning at the level 0 this name, level 1, 2, 3, 4, 5, 6. I'm assigning these name at different levels. So now I already assigned it and saved it. Now I am exchanging these names with these levels 0, 1, 2, 3, 4, 5, 6, 7, 8. We're using this command. I did exchange. Now I am renaming these command these name with these levels. So now I rename them by using this rename ID function, and let's visualize this in the form of plot. So before, what you have seen, you see the only the numbers. Now you see the labeling. So now you can clearly identify, okay, this is the naive T cell, this is memory CD4, this is CD8, this is NK cell, this is B cell, this is monocyte, CD14 monocyte, and dendritic cell and platelets. So now you have annotated these, all the cells. So this is the final step for the basics of single cell sequence analysis. Actually, these all the steps, how simple they look, they are not as simple as I show you. So there are more complexity in between, and the very complex thing here is this annotation of these cells. This is the very complex thing. And another complex thing is finding the number of clusters. How many clusters are there? How many clusters you want? So when you will learn this single cell sequence analysis, you will learn everything by time. Okay. So if you want to save this, what you have done here, you can save it by using the save RDS command. You have to put here your Swift object and you have to give the path. So this RDS command, your object will be saved in the RDS format. And whenever you want to resume the analysis later, you can just call or import your RDS file and you can start from the same level what you have stopped before and save it. Okay, I think this is the only uh, hands-on I want to show you for this session as an to give you just an example, the basics of single cell data analysis, how you can do it, what type of data you will get it, and how you can do the analysis, which package you will use. But of course, if you want to do this analysis, single cell analysis, you should have a knowledge of R programming. I can say at least basic knowledge of R programming. Otherwise, if you have a basic knowledge of R, then you can simply follow the pipeline and tutorials. And if you have a knowledge of Python, then you can follow the another pipeline called ScanPy. Yes. Okay, I will stop here and I will look the questions. If you have any question, let's check. Please repeat the sample data set. Okay, sorry, you can, sorry, I missed this, but you can download it again. Don't worry and follow the steps. If you will see, if you will tell me, ask me, I can also provide you the script I read, right? Just write, repeat the downloading. Okay, can you upload sample? Okay, I don't know if we can also upload here. But yeah, if you have looked, if you have followed my yes, you can write it. can we get the session? This question for Dr. Amix. Share to individual. Okay. Get it R code. Yes, I will provide you the R code for practice and also the sample data. Hope you are enjoying. Okay. Okay, I request Dr. Amis to share them attendance form and to do their other formalities while I'm taking the question. So because we are running with the time, we have only 10 minutes before ending the session. Okay, there is a technical question. If we have done a single cell RNA sequencing of my own sample and have raw data from the machine, how to get the initial file? Okay, so if you have done the single cell sequencing and you have a raw data, so one step have I have missed here, I can say, but that is not the part of this analysis. So if you get the raw data from the machine, actually for making it to the readable form for the student object, you have to do the processing of data or you have to do the alignment of data. And that alignment will be done using the Cell Arranger software, or it's an open source tool, you can say, not a tool, but is a software which you have to do for the alignment of single cell data. So data from the company or data from the machine, you will get in the very raw format. It will be in the FASTQ format. Then you have to align the data with if you say that if your data is human data, then you have to align the data with your human database, which you will find on this Cell Ranger package website. You will find those reference databases over here. So what you have to do, if you get the raw sequence data in the FASTQ format, then you have to do the alignment of that data. And once you run the Cell Ranger for alignment, you will get the output file in the form which I have loaded just now in this example. Okay. Okay, where will be the data? Okay, this is another question. So, okay, okay, yeah, I don't, yes, initial date, platform specific. Okay, someone has answered something. Alumina about these files contain millions of short. Okay, this is an answer. Can we get the, yes, and we will give you the R code. Will you get the recording? Yes, recording, I think we will get it. Okay, let's see if there is some technical question. I think now there is no technical question left in the chat box. And if you have any other question, any technical question, you can write to Dr. Omix. They will assist you. Okay. Okay, great. Yes, there is someone can use TCN. Yes, of course, you can TCN instead of UMAP. It depends upon which type of plot you prefer, which type of plot you like, but I like UMAP actually, because the visualization of this UMAP plot is great, which is very near to my heart. I can easily separate the cell type in UMAP, which you cannot do in the TCNE plot. Okay, great. Yes, again, this question was repeated. Explain my question. I asked if we combine, combine different data, do we have to make it need to count batch effect? Yes, of course. So this is a very technical question. So the question is that if we combine different data set, do we have to take into account batch effect? Yes, of course. So if we have sequenced the data with a different sequencing machine or at different time point with the same sequencing machine, then there will be a chance that there will be a batch effect. So to remove those batch effect, we can do the first one step like doublet finder. And doublet finder, I think, is even capable to remove the batch effect over with these if you have multiple data set. Then will this data will be processed? If yes, then hopefully, yeah, processed, yes. Not with omics, if I want to be a family. Okay, there are different packages. Like you can, if you want to do omics analysis, you can go to the simply the edge. You can do if you want to do the bulk RNA sequence analysis, you can do with the EdgeR, or there are different packages also available. DAXSet packages, EdgeR packages. You want to do single cell, I already told you, you can do with this. Uh, SWIRET, you can do with ScanPy. Yes.